otomatis.
Hidran yang berfungsi juga untuk memompa air secara otomatis.
Dan kami juga melengkapinya dengan smoke detector yang berfungsi untuk mendeteksi secara dini adanya segumpalan asap yang berasal dari api sehingga dapat menghindari terjadinya bencana kebakaran yang lebih besar.
Serta kami pun melengkapinya dengan apar, alat yang digunakan untuk memadamkan api atau mengendalikan kebakaran kecil.
Bagaimana jika terjadi keadaan darurat? Kenali tanda bahaya. Ada dua tanda bahaya yang perlu Anda ketahui: gempa bumi dan kebakaran. Jika terjadi kebakaran, harap tenang dan jangan panik. Jika mampu, gunakan alat pemadam api yang terdekat. Amankan dokumen penting dan barang berharga Anda.
Matikan aliran listrik dan tutuplah pintu untuk mencegah luasnya asap dan api.
Ikuti instruksi petugas kami untuk segera keluar ruangan melalui tangga darurat. Jika asap sudah memenuhi ruangan, segera Anda menunduk dan merangkak untuk menghindari asap menuju pintu keluar darurat. Apabila terjadi gempa bumi, harap tenang dan jangan panik.
Lakukan metode drop cover hold. Jatuhkan badan Anda dan lindungi badan Anda hingga gempa meledak.
Jika gempa sudah mereda, ikuti petugas untuk menuju ke titik berkumpul.
Segera lakukan prosedur evakuasi dengan benar dan tertib. Dahulukan ibu hamil, lansia, anak-anak, dan penyandang disabilitas. Jika terjadi huru-hara, harap tenang dan jangan panik. Tetaplah berada di ruangan sampai menerima instruksi dari petugas keamanan untuk meninggalkan ruangan. Tutup dan jauhi semua jendela. Bila diminta untuk meninggalkan ruangan, biarkan lampu tetap dalam keadaan menyala dan matikan semua peralatan yang ada. Ikuti prosedur evakuasi sesuai dengan arahan petugas. Amankan barang-barang berharga berukuran kecil dan dokumen penting lainnya di tempat yang aman. Saat terdapat korban henti jantung selama berlangsungnya aktivitas, minta bantuan orang lain. Untuk menghubungi tim medis reaksi cepat Rumah Sakit Umum Nasional Dr. Cipto Mangunkusumo pada nomor yang tertera di layar atau 0811 157 700.
Perlu kami sampaikan bahwa hari ini tidak ada pengujian alarm atau simulasi keadaan darurat. Jika ada alarm berbunyi dan ada perintah evakuasi, maka itu adalah kejadian yang sebenarnya.
Segera tinggalkan ruangan, keluar melalui pintu darurat, dan dilarang menggunakan lift. Ikutilah prosedur yang telah disampaikan. Petugas kami akan mengarahkan Anda ke titik kumpul. Tetap tenang dan ikuti arah evakuasi dengan berjalan cepat dan jangan berlari. Jika Anda memakai sepatu berhak tinggi, lepaskan sepatu Anda. Prioritaskan penyandang disabilitas, ibu hamil, lansia, dan anak-anak terlebih dahulu.
Pada saat di titik kumpul, petugas akan mendata ulang jumlah karyawan dan pengunjung yang ada. Petugas akan mendata ulang jumlah karyawan serta pengunjung yang berada di luar gedung sesuai dengan catatan yang ada.
Dahulukan keselamatan dan kesehatan Anda dalam beraktivitas. Demikian instruksi keselamatan dari kami. Terima kasih atas perhatian Anda dan selamat beraktivitas.
To all the audience, please be seated. The program will be started shortly.
Assalamualaikum warahmatullahi wabarakatuh.
Good morning, ladies and gentlemen. On behalf of the committee, we warmly welcome and sincerely appreciate the presence of honored guests. From Faculty of Medicine, Universitas Indonesia, FMUI, we would like to welcome the head of doctoral program in Biomedical Sciences, Professor Dr. Rerum Naturalium Asmarina, MS, head of the master's program in Biomedical Sciences, Professor Dr. Wawaimuli Arozal, M.Biomed, PhD. Secretaries of the doctoral program in Biomedical Sciences, Dr. Robiatul Adawiyah, M.Biomed, spesialis parasitologi klinik, subspesialis mikologi konsultan. Secretaries of the master's program in Biomedical Sciences, Dr. Rani Wardani Hakim, SSI, apoteker, M.Biomed. To all the head of department in FMUI, all lecturers and academic staff of the postgraduate programs in Biomedical Sciences, FMUI.
From University Kebangsaan Malaysia, UKM, Dean of Research and Innovation, Faculty of Health Sciences, Profesor Madya Dr. Satirah Zainal Abidin, Professors at UKM, Professor Dr. Nur Azizah, Muhammad Zin, and Professor Dr. Nur Fadilah Rajab. Lecturers at UKM, Associate Professor Dr. Arimi Fitri Matludin, Dr. Nurul Farhana Jufri, Dr. Izatus Shimataib, Dr. Farah Wahida Ibrahim, Dr. Siti Fatiyah Masre, and Dr. Mazlizam Abdul Latif. Also all keynote and invited speakers, researchers, and fellow postgraduate students. Welcome to the Postgraduate Collquium 2025.
Translational biomedical research, bridging the gap from bench to bedside. It is truly an honor to have you all with us today, and we hope that this colloquium serve as a meaningful forum to exchange knowledge, strengthen collaborations, and further advance the impact of biomedical research.
This prestigious event is proudly held in collaboration between the postgraduate program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, FMUI, and the Faculty of Health Sciences, University Kebangsaan Malaysia, UKM.
My name is Shinta.
And my name is Sylvie.
And we are deeply honored to be your hosts for this two-day colloquium here at the FMUI. Hopefully this will be a platform for sharing insights, strengthening academic partnership, and fostering innovations in the field of biomedical research.
Ladies and gentlemen, please rise for the national anthem. Indonesia Raya. Thank you. Please be seated.
To open our ceremony with the greatest gratitude, please welcome Zulfikar Isnain Akbar for opening prayer.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, on this occasion, I will lead the prayer in accordance with Islamic tradition. Please feel free to adjust accordingly. For fellow Muslims, let us raise our hands in prayer. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wa bihi nasta'in wa ala umurid dunya waddin. Wassalatu wassalamu ala asrofil anbiya wal mursalin. Wa ala alihi wa sahbihi ajma'in amma ba'du. In the name of Allah, the most gracious and the most merciful. Praise be to Allah, the Lord of the world, the master of the after, the creator of everything in this universe. Ya Allah, on this bright morning, we gather before you in both times of ease and hardship. Therefore, we ask you to accept our prayers and hopes. We thank you for granting us the opportunity to gather here today. Bless this event with your divine guidance. Make everything run smoothly and beneficially for everyone involved. Open our hearts and minds so that we may gain valuable knowledge and broaden our understanding. Ya Allah, please protect us from worry, from all harm and danger, from misfortune, from misery, and from calamity. Lead us always on the straight path. Grant us sincerity in our intentions and barakah in our time. Ya Allah, we know you are the best protector. Rabbi zidni ilma warzuqni fahma waj'alni minas solihin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar. Subhana rabbika rabbil izzati amma yasifun wa salamun ala mursalin walhamdulillahi rabbil alamin. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for the beautiful prayer, Zulfikar.
And now, we would like to invite the chairperson of the committee to deliver the speech. To Dr. Robiatul Adawiyah, M. Spesialis Parasitologi Klinik, Subspesialis Mikologi, Konsultan, please come to the stage.
Distinguished guests, respected faculty member, especially Prof. Arif Fahrial Syam as our Dean in FMUI, esteemed researcher and dear postgraduate students, and also honor participants. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi and also very good morning to all of you. It gives me immense pleasure to welcome you all to the Postgraduate Collquium 2025 with theme Translational Biomedical Research, Bridging the Gap from Bench to Bedside. This event is a proud joint initiative of the Master and Doctoral Program in Biomedical Sciences, FMUI, and the Faculty of Health Science, University Kebangsaan Malaysia. Today, we gather not only to present and exchange knowledge, but to reaffirm a shared vision, one that recognizes the essential bridge between scientific discovery and clinical application. In this era, where innovation in biomedical sciences moves rapidly, the challenge lies not only in advancing research, but in ensuring that our findings are translated into real-world solutions that benefit to the patients, healthcare system, and also society at large. This colloquium represents more than academic gathering. It's a platform for dialogue, collaboration, and transformation. This research presented here in this colloquium, from fundamental molecular studies to clinical insights, demonstrate the depth and the breadth of inquiry being pursued by postgraduate scholars. We also take this moment to celebrate the spirit of cross-border academic partnership. The collaboration between Universitas Indonesia and University Kebangsaan Malaysia stand as a powerful example of how regional cooperation can advance global health goals. Together, we foster a community of scientists who are not limited by geography, but united by a commitment to progress, integrity, and impact. To all participants, may this colloquium ignite new ideas, foster meaningful mentorship, and inspire pathway that lead from the bench to the bedside and beyond. On this occasion, I report that participants not only came from Faculty of Medicine Universitas Indonesia or also University Kebangsaan Malaysia, but also from other faculty in Universitas Indonesia and some other universities in Indonesia. There are 45 abstracts, 35 of them will be presented orally, and 10 of them will be presented to posters. On behalf of the organizing committee, I extend our deepest appreciation to our distinguished speakers, dedicated mentors, and all those who have contributed to making this event a success. Thank you, and I wish you all an insightful and inspiring colloquium. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih.
Thank you, Dr. Ada, for such an inspiring speech. The following speech will be given by the Dean of the Faculty of Medicine, Universitas Indonesia, to Profesor Dr. Ahri Fahrial Syam, spesialis penyakit dalam konsultan gastroenterologi hepatologi, MMB. Please come to the stage.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Associate Professor Dr. Satira Zainal Abidin, Dean of Research and Innovation, Faculty of Health Sciences, University Kebangsaan Malaysia. Okay. And also we have Professor Dr. Norazia Muhammad Zain and Dr. Maslizam Abdul Latif, speaker from UKM. And also we have our esteemed judge from UKM, Dr. Izatus Sima Taib, Dr. Nurul Farhana Zupri, and also Professor Dr. Nur Fadila Rajab, Dr. Farah Wahida Ibrahim, and Dr. Siti Fatia Marsre, and also the head and secretary of Master and Doctoral Biomedical Science Program from Faculty of Medicine University of Indonesia, faculty members, researchers, students, and colleagues. Selamat datang di FKUI. Yeah, I think first and foremost, let us offer our gratitude to Almighty for allowing us to gather here for the Postgraduate Collquium 2025, collaborative event between the Master and Doctoral programs in Biomedical Sciences at Faculty of Medicine and also University Kebangsaan Malaysia. And I think we also have programs, 3 university, UKM, UI, and Brunei Darussalam, I think every 2 years, to, every members to present research and also, yes, I don't know now, before Indonesia and then Brunei and then back again to UKM, something like that. I remember first time I went this meeting in UKM maybe around 20 years ago. So actually between us, between UI, UKM and Brunei, because using the name of the country, the name of the country of every members, we have joint meeting actually every 2 years. So I must check it is still going on or not. Yes, of course. Yeah, I think as a dean, the theme of the Translational Biomedical Research, Bridging the Gap from bench to bedside, I highlight the importance of turning discoveries at laboratory bench into tangible benefit for clinical practice and public health. We used this building actually from 2017. And most of them is the translational biomedical research. And yeah, hope so, not only the program, something like this, but also in the future we can make joint research because we have facilities and you have the facilities, so we are working together. I just give information, next month we have a meeting, the Dean of ASEAN, what we call that, ASEAN Medical Dean Network, so maybe I will give report about this important meeting because we always say that, okay, what kind of collaboration between Faculty of Medicine around region, especially for ASEAN countries. I don't know, I'm not mistaken. Sorry, this is from the Faculty of Health Sciences. Yeah. I mentioned that the Dean of Faculty of Medicine. Sorry about that. So I know that the Dean of Faculty of Medicine, Professor Nephrologist, I think still him. Oh, no, changed. Oh, pediatrician. Okay, maybe changed. Yeah, yeah. Sorry. Oh, Deputy Vice Chancellor, UKM. Professor, which the? Oh, Prof. Hanif, yeah, nephrologist, right? Oh, now up to. Oh, okay. Okay. Oh, good. Okay. Please say hello to him if you meet him. Okay, so again, to all participants, particularly our postgraduate students, I encourage you to engage actively, be open to feedback, and see this colloquium as a stepping stone for your academic journey and research advancements. And there is so many gardens around here. So also we have the house of orchard over there. And then please take your photo in front of our garden. And don't forget, as soon as possible, upload in your social media. Thank you very much. Okay. Okay, again, and last but not least, I will give pantun. I know that we have actually same language, so I will say pantun in Bahasa. Hope so you know about most of the word that I mentioned. Mentari bercahaya sangat cerah. Okay. Menyinari bunga-bunga di taman yang sedang merekah. Selamat untuk kolaborasi yang semakin meriah. Semoga kerjasama ini membawa berkah. Yeah. And I know that Friday you want to go to Bandung, right? Using the bullet train, that's right? Okay, and then hope so that you can find many things discount over there in Bandung. Yeah, many things. You can buy. You must buy everything from there. Yeah, uh, because you know that Bandung is one of my hometown as well. Actually. Okay, again, thank you very much. Thank you for your support and thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Ari, for the amazing and inspiring speech, also beautiful pantun.
And now, as much as we are going to remember this event, we want to preserve this by taking group picture. So we cordially invite from FMUI, please come to the stage, Prof. Ari, Prof. Asma, Prof. Wawai Muli, Dr. Ada, Dr. Rani, and all the head of departments from Faculty of Medicine, Universitas Indonesia, also all lecturers and academic staff of the postgraduate program in Biomedical Sciences, FMUI.
And from UKM, Profesor Madya Dr. Satirah Zainal Abidin, Profesor Dr. Nur Aziah Muhammad Zin, Profesor Dr. Nur Fadilah Rajab, Associate Professor Dr. Arimi Fitri Matludin, Dr. Nurul Farhana Jufri, Dr. Izatus Shima Taib, Dr. Farah Wahida Ibrahim, Dr. Siti Fatiyah Masre, and Dr. Mazlizam Abdul Latif.
Please come forward for the photo session.
Ladies and gentlemen, before we begin with our plenary sessions, allow us to briefly walk you through the agenda for today. After this opening ceremony, we will continue with 2 plenary sessions featuring distinguished speakers from Universitas Indonesia and University Kebangsaan Malaysia, followed by Q&A session.
And after a short morning tea break and entertainment, we will enter our first rapid-fire session where postgraduate students will present their research concisely and dynamically in parallel rooms.
Around noon, we will break for lunch, during which there will also be a poster display waiting for you to visit. You can also visit these posters display throughout the event.
In the afternoon, we will resume with rapid fire session 2, and later we have a special laboratory tour arranged for our guests from UKM, led by our faculty members and also the student committee.
We hope that today's program will run smoothly and be inspirational, insightful, and become a valuable opportunity for networking and collaboration.
To all the participants in rapid fire sessions and also for poster sessions, we wish you the very best. This is an opportunity to showcase your hard work, share your findings, and receive constructive feedback from fellow researchers and esteemed faculty.
Ladies and gentlemen, we are now about to begin plenary session 1. This session will be moderated by Professor Dr. Wawaimuli Arozal and Biomed PharmD. Allow us to briefly introduce our moderator. Professor Wawaimuli is a faculty member at the Department of Pharmacology and Therapeutics, Faculty of Medicine, Universitas Indonesia, where she has been actively involved in teaching and research since 1998.
She currently serves as the head of the master's program in Biomedical Sciences at the Faculty of Medicine, Universitas Indonesia, for the 2023 to 2027 period.
Her academic journey began with a Bachelor of Medicine degree from the Faculty of Medicine, Universitas Indonesia. She then earned her Master's Biomedical Sciences at the same faculty, with a thesis focused on enhancing the anti-inflammatory effect of methylprednisolone using liposomal delivery.
In 2011, she completed her doctoral degree, PhD, in clinical pharmacology from the Faculty of Pharmaceutical Science at Niigata University of Pharmacy and Applied Life Sciences in Japan. Her dissertation explored the protective effects of beta blockers and angiotensin receptor blockers against chemotherapy-induced toxicity.
Professor Wawaimuli's expertise includes pharmacology, cardiovascular diseases, herbal medicine, and oncology-related research. She has authored more than 180 scientific publications, especially in reputable Q1 and Q2 international journals.
Beyond academia, she serves as an assessor for the Indonesian Accreditation Agency, LAMPTKES, and is an active member of several professional organizations, including the Indonesian Medical Association and the International Union of Pharmacology, IUPHAR.
She is also involved in clinical trials as both principal and co-investigator, contributing directly to evidence-based practices in Indonesian medicine.
Her dedication to research and education has greatly contributed to the development of future scientists and clinicians in Indonesia. Without further ado, please welcome Professor Wawaimoli to moderate this plenary session.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all participants. Good morning to our guests from University Kebangsaan Malaya, University Kebangsaan, professor and lecturer, and also professor and lecturer from Faculty of Medicine, Universitas Indonesia. In this session, we have two distinguished speaker, one from University of Indonesia and one from UKM. So the first speaker I will like invite Professor Melva Luisa to present his lecture related to vitamin D. Times is yours, Prof. Melva. So you have about 20 minutes to deliver your speech.
So good morning, ladies and gentlemen, professors, doctors, and colleagues, and also our dear students. So good morning, and thank you for allowing me to present our research results entitled "The Role of Vitamin D in Prediabetes: A Multi-Target Approach to Inflammation, Insulin Resistance, and Gut Dysbiosis." So we know that the vitamin D is a fat-soluble vitamin, and we also know that its role to modulate calcium and phosphate homeostasis, right? So, but then we know that it has also the pivotal influence on immune modulation and also metabolic health. Its other extraskeletal functions include the cell proliferation, muscular functionality, skin differentiation, reproduction, and also safeguarding against the insulin resistance, etc. And the deficiency of vitamin D3 in our body will implicate in many abnormalities and disorders, which includes the various cardiometabolic and inflammatory diseases, which will be discussed this morning. And we know also that when the pandemic starts in 2020, right? So everybody starts to consume the very high doses of vitamin D after the COVID-19 pandemic. So this is the metabolism of vitamin D, which the UVB will convert the dehydrocholesterol in the skin, in our skin, to provide vitamin D, and then it will transform the vitamin D with heat. And then it will enter the bloodstream, binds to the vitamin D binding protein, and then transported to the liver and undergo hydroxylation. And in the liver, it will result in the formation of calcidiol, or 25-hydroxy vitamin D3. And then it will subsequently converted into the active metabolites, which is the calcitriol, and the synthesis is undergone in the kidney. And the synthesis of calcitriol is influenced by the parathyroid hormone stimulation, but it is inhibited by the high levels of calcium, phosphate, and the high levels of the calcitriol itself. And this is why everybody starts to consume the vitamin D in very high doses, that its modulatory effects. And how the vitamin D have immunomodulatory effects is due to the both calcidiol and calcitriol exerted its immunomodulatory effect by regulating the function of macrophages and dendritic cells, the two key players in the immune system. And it also affects the T cell responses by inducing the dendritic cells. Thus, it will influence their ability to combat infections, regulate inflammation, and initiate immune response. Sorry. So this is what we are going to discuss today, the pathway in which vitamin D3 deficiency leads to insulin resistance, prediabetes, and finally to type 2 diabetes. So the vitamin D deficiency will hampers the insulin signaling pathway by decreasing the GLUT4 and insulin receptor expression. Thus, it diminishes the calcitriol anti-inflammatory effects, which ultimately resulting in insulin resistance, subclinical inflammations, and it negatively affects the insulin synthesis and secretions to both genomic and non-genomic actions. And this is how the progression from insulin resistance to beta cell dysfunctions, which is followed by metabolic changes that lead to complications. And actually the process will range from 5 to 10 years. And since the prediabetes is identified as a pivotal juncture, and then because many evidence suggests that this condition is still reversible at this stage, so many researchers said that this point might be a feasible avenue to prevent the onset of diabetes mellitus. Then the strategies aimed at reversing prediabetes will include so many things, including lifestyle modifications, pharmacological interventions, and also the vitamin D supplementation. Yeah, sorry. Yeah, so actually this is the gap, yeah. Since many literature has identified that the serum vitamin D levels is a significant factor as a progression from pre-diabetes to type 2 diabetes, it merits the considerations that it can be a strategy for preventative and interventional strategies. However, the existing literature presents inconsistent findings. Emerging studies indicate that there is inverse relationship between the level of vitamin D and prediabetes risk. However, there are some studies also found there was no association between the serum levels of vitamin D and the risk of diabetes. Therefore, to address this gap, we conducted 2 studies, a cross-sectional studies in a high-risk community and an experimental study in a pre-diabetic rats. Yeah, this is the study just before the pandemic hit us. Yeah, we did a study of community, and we did it as a part of community engagement project. Yeah, so this is what we considered at a high-risk community because they have mainly air pollution, poor housing conditions, minimum ventilations, so that they have a limited sun exposure, and then there are also unhealthy heating habits there. So we did a study to investigate the relationship between the levels of vitamin D with the markers of metabolic syndrome, as well as the glucose metabolic control markers. And we sampled about 221 subjects here. Okay, so this is what we found. Yeah, so we know that the world has already seen the vitamin D deficiency as a pandemic, yeah, because the global condition of vitamin D deficiency is right now is about 40 to 50% in the population. But in our study in the population, we found a very high deficiency of vitamin D of less than 30 nanogram per milliliters. It's about 74% that have low levels of vitamin D, and which the 40% are below 20 nanogram per milliliters. And this is what we found about the glucose control conditions. Then we found that based on the glycated hemoglobin, we found 14% of undiagnosed type 2 diabetes mellitus based on HbA1c, and so the HbA1c is more than 6.5%, and 24% of undiagnosed prediabetes with the level of glycated hemoglobin between 5.7 to 6.5%. But when we do a correlation analysis between the level of vitamin calcidiol with the HbA1c, we only found a positive weak correlations between the two. And what about the status of vitamin D in undiagnosed diabetics and pre-diabetic subjects? This is the trend that we found, but we failed to see the statistical significance between the level of vitamin D, the low levels with the risk of diabetes and pre-diabetes. And then to try to understand how the vitamin D supplementation works in pre-diabetic conditions, we did the second study, which is to investigate the mechanism of action of vitamin D3 given as supplementation on pre-diabetic rat models. So we did and we induced a rat model with a high fat, high glucose and streptozotocin. One of the group was the healthy rats, the first one given the healthy diets, and the second one was given high fat, high glucose and streptozotocin diets with untreated, so no medication was given. But the third and the fourth group, we give supplementation of vitamin D. The third group, we give us vitamin D of 100 international unit per kilogram per day. And the fourth group is the 10,000 international unit per kilogram per day. The doses is a little bit similar to 500 international units and the 5,000 international units. So the high doses that people are trying to consume during the pandemics, right? Yeah. And then the 500 is the over-the-counter medications that we can see on the market. Okay. Okay, so this is our first result. Yeah. So our first result regarding the findings of insulin resistance and glucose control parameters, we see that actually in pre-diabetic and normal condition rats, they have a similar baseline of vitamin D of about 25, yeah, but all are below the 30 cutoff point of normal levels of vitamin D. But then if we try to do a supplementations on both, the normal or the pre-diabetic conditions, they tend to have a reduction in vitamin D levels in their body. But then if we give supplementation, it will lead to the increase in about 2 to 3 points of nanogram per milliliters in 12 weeks. And then when we do a histopathology analysis, we see that the vitamin D will minimize the damage in the Langerhans islets and then reduces also the fatty pancreatic cell atrophy. Yeah. Regarding the insulin resistance markers, we see that the vitamin D3 will reduce insulin resistance and provide better parameter for glucose control than the untreated rats. But if we see the low dose and higher dose, we only see a minimum difference between the two groups. Okay. Yeah. And then in the molecular level, we see that the vitamin D3 treatment will reduce the phosphorylation of IRS1 in pre-diabetic rats, but only causes a slight changes in PPAR gamma expression, but reduces the NF-kappa B expressions. And then if we look at the inflammatory markers, if we see the individual markers, we only see small changes in the IL-6, IL-10, also the TNF alpha. But if we are looking at the ratio of EL6 per EL7, then we will see a really good reduction in the inflammatory markers. Yeah. And then we also see that our model of pre-diabetic that we induce with high fat, high glucose, and streptozotocin resulted in the obese rats, yeah. And then we see that in the vitamin D3 supplementation, both in low and high dose, it causes to normalize in the body weight, Lee index, and all of the body weight parameters. We also see that there is a reduction in adipocyte cell levels in the pre-diabetic rat model. And then we can also see that there is a reduction in adiponectin per leptin ratio, but we only see in the we only see a reduction in the leptin ratio, but not in the adiponectin. Yeah. Yeah. Regarding the diversity on the composition of microbiome, we see that the vitamin D3 supplementation can modify the composition of microbiome at phylum and genus levels. Regarding the richness of the species and the diversity of the gut microbiome, the vitamin D3 will be able to modify the composition of the microbiome, both at phylum and genus levels. But if we can see at the low and high concentrations, there is no marked difference between the two. Okay. And this is what we found in the kidney. Actually, in pre-diabetic models, we have already seen a marked reduction in kidney function markers, yeah, urea, creatinine, albumin, and also KIM1, kidney injury molecule 1. But then no changes were caused by the supplementation of vitamin D in terms of urea, creatinine, and albumin. However, if we see it on the kidney injury molecule 1, we can see a reduction given by the high dose of vitamin D3 supplementation. So this is how we summarize our findings regarding our experiment in the pre-diabetic rat model. Yeah. So vitamin D supplementation can demonstrate improved glucose metabolism, mostly by the reduction of insulin resistance and preserving the pancreatic islet structures. Yeah. And the adipocyte, it will reduce the adipocyte size, and then also reducing the ratio of adiponectin per leptin ratio. And it will cause the reduction in inflammations, and then supporting the expression of PPAR gamma in IRS1, so it will become reduction in insulin resistant in the rats. So here are our conclusions out of the 2 studies that we did. So actually the pandemic of vitamin D3 deficiency in high-risk communities, the communities with the air pollution, limited sun exposure, unhealthy eating habits is significantly higher than it is recorded in the prevalence, and its correlation with HbA1c actually has been identified and linked to pre-diabetic and diabetic populations. However, this relationship is still need to be clarified and done in a strict methodology manner. Yeah, and our experimental pre-diabetic animal model, vitamin D3 supplementation has also been shown to improve insulin resistance, and which may be attributed to the decreased IRS1 phosphorylation, reduce inflammatory markers. And also it decreases obesity in pre-diabetics by restoring the adiponectin per leptin ratio and modifying gut microbiome composition at the phylum and genus levels. And this can be, I think all of the positive and negative, all the things that we found still supports the use of vitamin D supplementation in pre-diabetics to prevent the progression of insulin resistance to diabetes. And then I would like to acknowledge all of the team that do the study here. I think we have several researchers here, maybe Ibu Desak and Prof. Ernie is here in the room, supporting and then doing the study. So with that, I thank you.
Thank you very much, Prof. Melva, for a very interesting lecture. So she already talked about the relationship between vitamin D deficiency and the pre-diabetic condition. So Prof. Melva already share about the two kind of study. One is a research study related to cross-sectional study in community with high risk of vitamin D, and the other study is in experimental study using pre-diabetic rat. So before we further to the next presenter, I would like to show the CV of Professor Melva, please. So sorry, but we not yet show the CV of Prof. Melva first. Yeah. Yeah, so this is the curriculum vitae summary of Professor Melva. She's one of the staff of pharmacological department in FMUI, and also, he's completed her PhD in biomedical science in Faculty of Medicine. Okay, next presenter is Dr. Please show the CV of Prof. Nur Aziza. Yeah. The next speaker is Professor Nur Aziah binti Muhammad Zin. He's a lecturer, professor from Faculty of Health Sciences, University Kebangsaan Malaya. So please see the curriculum vitae of Professor Nur. So time is yours to present your lecture. Thank you very much. So you have 20 minutes for presentation.
Right. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Madam Chairman. Thank you very much for the organizer for inviting us to be here. It's very pleasure for us to be in the UIM. And then we are from delegates from University Kebangsaan Malaysia in Kuala Lumpur. So we have about 9 members in our team. Right, so what I'm going to do this morning is to present the work that has been done in our lab. Is collective works about the antimicrobial resistance bacteria, especially that we are going to sharing maybe the general knowledge. Maybe it's good for the postgraduate student that involved in this area, as well as for the future collaboration that we establish the method, the assay in our lab. Right. So as for the introduction, all right. As you know, the AMR, or the antimicrobial resistance, is ability of the microbes, including the virus, fungi, viruses, to resist the effects of the antimicrobials. For this talk, we're going to specifically concentrate on the bacteria for the antibiotic resistance. So for these particular problems, is make the treatment make it difficult to treat the infection, even though it's common infection in the hospital. And mainly for these cases, a lot of hospital-acquired infection taking place among the patient. So this is involved of a lot of tasks, a lot of things need to be done in order to to get the appropriate treatment. Right, for these antibiotic things, we're starting with from the maybe early 18th, 19th century, or in Malaysia especially, we have starting with the natural product, turmeric, garlic, honey, and so forth. I'm sure in Indonesia as well, we have several traditional medicines, and up until now, people are using this as a supplement or adding value for the medicine that we have. Right, in the penicillin start discovering in the 1938. Right, for the bacteria itself, we are talking about the mechanism of action. We are going to very familiar with this terminology, the bactericidal and bacteriocytic. Which one is the bactericidal is to kill the bacteria, and then the bacteriocytic is the effect is just inhibit the bacteria, which maybe and then of course with the times going on, we have several antibiotics that has been produced in the pipeline. It's either narrow or broad spectrum antibiotics. So for this AMR itself, as I mentioned just now, is reduce the effectiveness of the treatment as long as prolonged hospital stay, and it costs a high medical cost. Right, in terms of history behind this, for the timeline from as early as 1904, starting discovery of for the treatment for the syphilis, and then the penicillin taking place is 1928. It is considered up to 1932 is considered as dawn of the antimicrobial, and from that time onwards until maybe early 1980s, we have the golden era of the antimicrobial. Several, about more than hundreds antibiotic has been produced, and then but bear in mind, in the 1960s, the first methicillin-resistant Staphylococcus aureus has been reported. And starting from that timeline, we have several resistance taking place. Lactamase producing organism that's been found in Germany, and then the vancomycin, Pseudomonas acinetobacter baumannii, and so forth. So there is there's the rise of the antimicrobial resistant phase. And then starting from that, we can a lot of resistance bacteria coming in, right? And then back to last 5 years ago, the WHO established the global response of the MR together with the Wellcome Trust. Okay, so with this WHO statement, AMR become is top of the 10 global public health threats. The top 10 is including other related diseases, and this is the biggest global threat in health in 2019. Right, so we go to the global burden of the AMR. As you can see here, it's speculated in the 2050 is going to have 10 million deaths worldwide, with this involve 100 trillion cost for the hospital stay, for the treatment for all over the world. So in this increased problems, increase of the cost is become the AMR is become a real challenge for us in the world, especially in the ICU patient. Right. So for the factor, maybe involve different factors for the rising this antibiotic. So this is including the overuse or misuse of antibiotics in human medicine, agriculture, especially in the livestock production. Yeah, that accelerate the development of resistant strain among the poultry, especially, and then pure hygiene and infection control, and global travel and trade. Right, so I think we can focus on the research that we have, and then all over the world, that we can do the many research involvement, a lot of different parties of different multidiscipline researcher to combat this problem. Yeah, right. And for the last 20 years, not many antibiotics or pharmaceutical company involved in this particular research because of the cost and very limited resources. So the discovery and the development is very slow. Yeah, and then of course, at that time the resistance developed very quickly. And then, of course, on top of that, the complex mechanism involved in this particular pathogen, then make it difficult to predict the resistant pattern as well. So in this particular view, with the global surveillance gaps, with the different mechanisms such as biofilm formation, make it the bacteria is more resistant towards the existing antibiotics. So in the whole area for the novel approach to combat the resistance, we have maybe 20 of the approach, but what I'm going to highlight here is maybe 4 out of this. We're starting with the antimicrobial peptides, which is the acronym for the AMPs. Yeah, so this is the one of the research that can be conducted, yeah, by using the AI, artificial intelligence, and using the machine learning, meaning to say that they are using the public domain of the data of the different peptide, and then targeted peptide, docking and undocking, and then it can be synthesized, the peptide, to be used for the bacteria purposes, for the antibacterial purposes. So as you can see here, they have several mechanisms, yeah, from the membrane disruption to the immunomodulatory effect. So with these particular peptides, with very short, consists of up to 50 amino acids, they can act very quick, and then in the very rapid and very short time in order to combat the pathogens. Right, so for this one example that we have here in our collaborator, that they use this AMP, antimicrobial peptide, to against the Klebsiella pneumoniae in the hospital, the clinical sample. So we are going to use the peptide, yeah, antimicrobial peptide that has been produced, meaning to say that they are synthesized this AMP, but to be able to be used in the lab, and after that in vitro and in vivo study, we have to test first. Right? Not necessary that all the AMP that produced in silico can be used or is active against a particular bacteria that we use in the lab. So meaning to say that maybe they have 5,000 synthesized AMP, and maybe 10 of that are good enough to good binding and so forth. And of course, out of 10, maybe 2 only can be used to actively kill or inhibit the targeted bacteria. Right, so of course they have some challenges as well in this particular research. Yeah, maybe in terms of stability, toxicity, delivery mechanism, and the side effect. Right, so for this particular AMP that been produced. Maybe we can start with the, let's say, Klebsiella pneumoniae. Yeah, we have the full sequence of that. So they have the targeted sequence. So and then the sequence that available in the database in the public domain can be used that comparing with the Klebsiella pneumoniae that we have in our own clinical setting, clinical sample bacteria. So that with the AMP that been synthesized, bring back to the lab and start doing the assay, in vitro assay. So that kind of experiment that we're going to do in the next few years. Right, for the example for the another research that been conducted in our lab is using the nanoparticles. As you know, the nanoparticle itself is very small molecule that have the broad spectrum activity, and some of them can give a very good synergistic effect. Yeah, right. In our case, we were using the nanoparticle itself, photocatalytic nanoparticle that coated to the PPE. Yeah, in order to give some antimicrobial effect to the PPE itself, so that it can protect the operator, the healthcare staff, while they are wearing the PPE. In this case, we use the surgical gown and surgical mask. And from the primary study, it shows that this photocatalytic nanoparticle, meaning to say that when exposed to the light, is a catalytic by the light exposure and give the ROS activity, and then it can kill or inhibit the bacteria. And it's found that it's between the fungi and the bacteria is more active against the both gram-positive and gram-negative bacteria. Another research on is also can be explore in this particular area is using the probiotic bacteria, yeah, lactobacillus especially, that has known for ages as a benefit to the host, yeah, and then is commonly found in the fermented food like yogurt, kefir. We have several fermented Malaysian originally food like what you call it, the budu, and then the what you call it, the cincaluk, the fermented food. Yeah? But one of the research that we carry out with the universitas in Padang, they have using the dadia. Yeah? Dadia is part, not dade, in Malaysia we call it dade, is totally different. In Indonesia, dadia is made from the buffalo milk. Yeah, so they use isolating the lactobacillus for the vagina bacteria. So it is ongoing project as well. So and then the published article in this particular area that we isolating the lactobacillus from the breast milk, and as well as from the kimchi is still in press for the publication. Right, so it's become more interesting if we can talk about the medicinal plants. Yeah, they have various phytochemical. I think many of my members working on this particular area as well, with for different activity. Yeah, for different activity. For myself, concentrated on finding the antimicrobial activity. Right, as we know that they have like alkaloid, yeah, terpenes, phenolic compounds from the plants that can be used to be good antibiotics against bacteria, and of course can be combined with the existing antibiotic as for the synergistic action. Right, in our group, we are producing this auracin A that isolated from these Basidomyces nanophanes nambi against the MRSA. So this is collaboration with the FRIM, yeah, the Forest Research Institute of Malaysia. And we found that this auracin A is we have the very good novel activity as compared to the existing antibiotics, and then against the methicillin-resistant Staphylococcus aureus. Right. For the another one is on this particular endophytic streptomyces. This is that's my what I call it my forte of my research, isolating the bacteria. Streptomyces the main producer for antibiotics nowadays. So we are isolating the bacteria from the plant. So whereas maybe we have about more than 200 isolate now. So for the past 20 years. So some of the projects still carry out. So we are using different methods, metabolomics, genomic approach in order to get the what you call it, the bioactive compound of it. So for the SUK48, Streptomyces SUK48, we managed to have to get about 11 antibiotics out of it for the biosynthesis, especially for the aura varantine, that is have a novel activity against specific bacteria. Is still under application for the patent, so I cannot disclose at the moment. So for the another isolate that we have is also against the Staphylococcus aureus, but with a different compound. And then in this case, as compared to the chlorophenol, they have a multiple biological target. Is much more better because it's as compared to chlorophenol, only have a single target to kill the bacteria. So we found that these genes, this particular bioactive compound, cycloval pro, is can regulate down-regulated genes for the Streptomyces, for the Staphylococcus aureus methicillin resistance. So another project that we were dealing with using the Malaysian stingless bee honey, as known as Kelulut, it's collaboration with University Technology Malaysia. So they have that mechanism using the different assay, different using the electron microscope to see the effect, how the Kelulut kill the the bacteria, yeah, by the interrupted to the septum formation and blocking the DNA and protein synthesis of the targeted bacteria. So we now go to the future, what you call it, the future outlook, yeah, the potential solution to combat antimicrobial resistance. As you can see from the beginning, yeah, this problem, yeah, this the problem arises because of the bacteria itself, the ecological, the evaluation, the mutation. So the technology has to be at par, has to be extensively at par in order to tackle or to solve the problem. Yeah, the diagnostic, the surveillance must be very good. Yeah, the biosensor technology, for instance, global data sharing initiative, maybe Malaysian data, Malaysian hospital data, and Indonesia hospital data on the AMR can be shared. Yeah, we have one ongoing project. One of my PhD students here, the project is about to use some device to sterilize the format in the hospital. But we end up with the first of all to see the baseline, yeah, the bacteria baseline in the hospital, and we end up with so many multi-drug resistant organisms in the format in the hospital. And very scary to say that, yeah, not only resistant to 2 or 3 antibiotics, but resistant to 24 types of antibiotics. On the format, yeah, on the format, like stethoscope, BP cuff, yeah, glucometer, thermometer. So this kind of things needs to have multidiscipline collaboration, not only from the physician, but from the researcher, from the people who talk, people who know how to conduct the research as well. Yeah, right. The targeting networks, yeah, with the existing drug, and then the revived bacteria susceptibility to existing antibiotic as well can be done. This program has been introduced from the WHO, the Global Action Plan, yeah, the stewardship and policy solution, how it can be prevented. Yeah, improve the awareness, yeah, strengthen the knowledge, must be through the surveillance and as well as the research, and the infection control unit in the hospital as well has to be play important role. Yeah, how to reduce the incident and infection, how we can use this disinfectant, yeah, using the different type of disinfectant which is rapid, which can be used for all type of format in the hospital, so it can reduce the transmission of the infection, right? Optimize the antimicrobial use is among the GPs and physicians, yeah? And then ensure the sustainability investment in this particular area. Future outlook, maybe we can see how it can addressing the AMR require a lot of innovation, awareness, and global cooperation. Maybe we can start with Malaysia, Indonesia collaboration on this AMR, or maybe it's already been established and already has been developed, but need to be strengthened. Yeah, invest in research, educate healthcare provider, and develop the sustainable and global policy. This is our team members from the students and from the other universities that involved in the project. With that, I thank you.
Thank you very much, Professor Nora. So please come to the stage, also Professor Melva. Now we will start for question and answer session. So I would like to invite the audience to raise a question to our lecture. Yeah, so we have about 15 minutes for question and answer session. So if, waiting for the question from the audience, I would like to ask to Professor Melva. Yeah, maybe because of the goal of this colloquium, we will create a kind of research collaboration between UKM and also FMUI. So what do you think about we can also create kind of cross-sectional study, yeah, to check whether the pattern of deficiency vitamin D in Malaysia also similar with Indonesia. So could you like to have any idea, yeah, how we can do the research together? Please, Prof. Melva.
Thank you, Prof. Wemuli. Yeah, it's very interesting that what we find in a cross-sectional study that in urban population where we think that there is a high risk of less sun exposure, and then they live in an urban population that's mixed with so many risk factors, that maybe we can check how many risk factors that will lead to the reduction in the vitamin D deficiency, and then we might be able to create something like what you did with the antimicrobial resistance with and then handbook and everything, maybe in the high-risk communities where maybe we can do that, and as part of community engagement also. Yeah. Okay, Prof.
Yeah.
Thank you very much.
So what is your opinion? Yeah, Prof. Yeah. Okay. Melva.
Thank you. Thank you, Prof. Milva, for a very nice presentation. At the moment at our faculty, we have this study on vitamin D status among office workers, whereby they are not exposed too much with UV or with sun exposure. And the study has already, for the cross-sectional, we have finished. That is the risk, the high risk population that we see among Malaysian, whereby their vitamin D level is quite low. Conducted by our nutrition lecturer, Dr. Aini Jamil, and I'm part of it. And now we are looking on how this also can lead to osteoporosis of the bone. And especially when it comes to the people with postmenopausal or premenopausal, whereby they are the one that already have been in the office, work in the office, not exposed to much of the sun. They are the high risk that will lead to bone health related condition. And we also look into different inflammatory markers as well, and their effect into aging and also DNA damaging effect of because they are not exposed to vitamin D and UV as well. Because absorption of vitamin D is actually very important. It's not just the level, but also how it is absorbed. So when they are not exposed too much on the UV, they will lead to low vitamin D and lead to osteoporosis. That is one that still conducted by our PhD student at the faculty. If you want to have the connection, we can provide the details later. Thank you, Dr. Velva.
Thank you, Professor.
Yeah, thank you very much, Prof. So next question from Dr. Ada, please. Yeah.
Yeah, for opportunity, thank you very much. For Prof. Melva, we are waiting for the clinical trial, yeah, Prof, to human, because this is very important and very interesting. Actually, I want to ask Prof. Nora one thing. We already know that AMR for bacteria is already done in some countries in Asia, Europe, and etc. But what do you think about the fungal? Because fungal, yes. Because WHO is already released about FPPL, right? Maybe you have experience about it. Thank you.
Thank you for the questions. For the fungal, we have the expertise in our faculty, Dr. Jacinta, but she's doing a lot of anti-fungal activity as well for the resistant among the fungus. But especially for the fungi cases, among the immunocompromised patient especially, the resistance is out of control. Among the immunocompromised, HIV positive, the cancer patient, once it's been infected by the fungus, is very, very difficult to handle. And many of the existence anti-fungi drug is not work anymore. And then I remember that they have the problem with the people nowadays, the young generation or maybe old generation have the cats in the house, as a pet. So many infection from fungal infection from the cats can be transmitted to human, and then they have systemic as well, and make the infection cannot be treated easily. And then some from the using of the essential oil, the research on the different type of essential oil as an antifungal is one of the field of study, field of research that you can explore. It's a lot of interesting finding is about for using the essential oil as an antifungal. Right. Thank you.
Yeah, thank you very much, Prof. Nora. Is there any question from the participant? Yeah. Yeah.
Before going to the, I
yeah.
want to address the maybe the collaboration ideas. Maybe I still have actually the samples, and then we are planning to do the vitamin D receptors polymorphisms, because we know that actually in Indonesia is very rich in sun, right? In Malaysia is also rich in sun, but then 75% of the samples that we take is have a deficiency of vitamin D, that we might we think that there is something with the receptor polymorphisms. So that's maybe, Prof, why we can do something in Malaysia. UKM can do the analysis of the genetic data, and then we have our sample also, and then we can combine it, and then we have samples, rich samples that maybe we can publish it with in a good.
Good impact. So for example, so you still have your biological sample, right? The blood, and then you can do some genetic analysis related to receptor. Yeah.
Yes.
I see.
We also still have we have over 200 samples that. Yes, maybe a difference. Yes.
Yeah, a very good idea. So next maybe we can have together create a proposal. Thank you very much.
Thank you very much, Professor.
Maybe next question from the, Dr. Rizky. Yeah.
Thank you very much for the opportunity from the committee, and also I would like to thank you for Prof. Melva, and also it's nice to meet you, Prof. Norazia. But I want to ask a question to Prof. Melva, because I'm focused on the degenerative disease model. Yeah, I also would like to ask Prof. Norazia, because I'm also a medical doctor, and for psych, AMR become one of the major problem, especially in the intensive care unit. So for my first question for Prof. Melva is, aligned with our theme today, bridging the gaps from bench to the clinical translation. We know that to create a results that is more translatable, we should create our project, especially in vivo, that is also mimic to the human condition. But I also want to see your insight and perspective, Prof. Melva, especially for us as a first, or maybe a freshman, we can say, a freshman as a researcher, to conduct a translatable researcher. For example, research. For example, like the models that you create, streptozotocin plus high fructose and high fat diet. I also have read that those models have some limitation, like you say that this model is a pre-diabetes models, but some other paper says that streptozotocin might reduce the pancreas cells, which is, some other research complain with that. Any other insight and perspective regarding this, Prof. Melva, so we can create a research that is more reproducible and more translatable? That is my question first for Prof. Melva. For Prof. Norazia, but this is just for my curiosity. One major problem that, based on my experience, occur in the hospital, especially in the intensive care unit, is the abusive use of superior drugs. For example, like there are a lot of intensive care doctor, but this is not a skeptical statement from me. I just seeing based on my experience, receive the vancomycin or meropenem and other, that it will lead into the antimicrobial resistance. And the major problem is, as like you say, that our research are very slow. My question is, in Malaysia, especially maybe in the regulatory level or maybe in your institution, how you tackle this based on your perspective, Prof. Norazia? Thank you very much. That's my question.
Let me answer. Thank you, Dr. Rizky, for the questions. The first one is how to do a translatable in vivo to human studies, right? Yeah. So actually, if we do a study in human, we try to do it as pragmatic as possible, so that when we translate it into the everyday use, it will be like the everyday what that we use. So if we design a clinical trial and then we put too much inclusion and exclusion criteria, when we want to translate, then everything will not be translatable, right? So that's how we have to design it as pragmatic as possible. But then the limitation is for a pragmatic trials, we need so many sample sizes, yeah, so that it will have the enough power to conclude. And then in times of efficiency like this, we need to think harder, yeah, to do a design of a clinical trial and a translational trial that can translate it into the metabolic conditions. Regarding the streptozotocin, actually it's not the best models to mimic the metabolic syndrome or the diabetic models, but the high fat, high glucose diets is actually, if we use it in a longer period, it will translate more into humans. But then we need about 6 to 12 months to do that, and we don't have that much time, yeah, to conclude, yeah. And streptozotocin that we use in our study is actually a very low dose, so it will not make damage in the pancreas so much. So we'll still see the healthy pancreas, only a slight damage, but then the vitamin D3 treatment are shown to reduce the damage, yeah. And then the one that can also be translated into metabolic syndrome model in animals, actually canteen diet. So yeah, cafeteria diet, yeah. So cafeteria diet is given to the animal, and yeah, for 3 or 6 months, yeah. But still, to do research that can result into a metabolic disease, we need a long time, yeah, to produce. And for students, it's actually not a very, yeah, maybe not for the master students, but the doctoral students may be okay, yeah. Okay, I hope that answers your question, Dr. Rizky. Thank you very much.
Right, Dr. Rizky, thank you for the questions. Right, regarding the for the ICU patient, yeah, the doctors or the specialists tend to prescribe the highest, strongest antibiotics for the patient. Yeah, as you can see from my presentation just now, is the misuse and the overuse of the antibiotics is critical to the patient, yeah, to that, what you call it, to increase the antimicrobial resistance, because you know the genes at the molecular level, the genes that carry the antibiotic resistant genes that been carried out by the certain bacteria can be horizontally transferable to the other bacteria as well, you know, and then prescribing the strongest antibiotics will kill all the other normal flora, and the patient, the sick patient, maybe become more sick. That's why for the laboratory people point of view, yeah, the AST result, the antibiotic susceptibility test result has been very accurate. So all this is all depends to the laboratory people, how they conduct the AST test, and then how they accurately report the patient from the patient samples, which antibiotic this patient still susceptible. It's very quick and very rapid in order to give the best treatment to the patient, not to use suddenly to use the strongest antibiotics, suddenly to use the broad spectrum antibiotics. So for the research point of view, maybe the device or the biosensor that we can develop. Because AST itself maybe takes overnight, 18 hours to get the result. But with the development of the biosensor, for instance, they use electric field that can be detect resistant genes very quickly, in 2 seconds, for instance. Of course, we have to refer back to the gold standard AST test, but with this very quick, in 2 seconds, you get that, all right, this sample contain the antibiotic, or this sample contain the resistant genes. So you can, particular resistant genes, so you can use a particular antibiotic to treat the patient. That's all, thank you.
Thank you very much, Prof. Nora, for your answer the question. So, is there any question from the participant? Yeah, I think I have one question to Prof. Nora. So, I am interested with your research related to antimicrobial peptide. Yeah, so you do the in silico research, yeah, to find some peptide that may be promising, yeah, to combat antimicrobial resistance. So in the future, do you think that you want to do the combination with the certain antimicrobial, or you do the separate? I'm not sure. I mean, what is your plan to do in the setting, in the clinical setting? Yeah, thank you.
Okay, thank you very much. Yeah, this is my new research that I embarked for, maybe just met Dr. Farid in FST. Right, but in silico, they developed by themselves. The bioinformatics, you know, from the science and technology, they screen all those potential peptide molecules and then doing the docking and docking. In my case, I have the whole range of the bacteria that we can be tested, the assay that we already established. So so this is very important for the multidiscipline. Yeah? We are not bioinformatics, surely. We are not the people who are doing the docking. We are the, maybe we know our field. So the combination of the people that doing in the same thing with the same interest, so it's make it a good research team. You know, so from that point of view, we get the sample. They already have the sequence, the peptide sequence. They send to the computer to synthesize that, and we use that to be tested in our lab. So in some cases, of course, for example, they try to combine with the tetracycline. Yeah, this tetracycline is out of dated antibiotic, in that sense that it's not many bacteria susceptible to them. So we combine the peptide, because peptide is very low molecular weight, combined with the tetracycline or other antibiotics that already been used. So it gives the synergistic effect as well. And then of course for formulation and for the first clinical trial, second clinical trial, we need other pharmacies or maybe people that involved in that area. Yeah, that's all. Thank you.
Very interesting idea. Yeah. I think, I mean, we don't have enough time to discussion, but maybe after this session, so we still have faculty meeting, business meeting, and then maybe our guests from UKM and then also the lecturer from University of Indonesia can discuss together to create a proposal together, yeah, to create research together, yeah, for the next. Okay. Thank you very much for the lecturer, for the nice lecturer, Professor Melva and Professor Nora, and then all for the participant. I would like to end this session. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for Professor Wawaimuli, Professor Melva, and Professor Norazia. Also to the participants who asked an incredible and insightful questions. And now I would like to invite our head of doctoral doctoral program for biomedical sciences, Professor Asmarina, to give a goodie bag as a token of appreciation for our speakers. And please stay for the photo session. What a wonderful plenary sessions from our professors.
It's time for a short coffee break. We also have entertainment prepared by our committee that showcases medley traditional dances performed by Sanggar Art Nusantara Production. Enjoy!
Saya
What a great performance by Sanggar Art Nusantara Production. I can only imagine the difficulties in deciding which dance should be performed because we have so many to choose from. And once again, let's give a round of applause for the performers.
We will now proceed with the rapid fire session 1, where selected postgraduate students will deliver concise presentations of their research. Kindly make your way to room 1 or room 2 as listed in the program booklet. For those who are listed in room 1, please stay in this room. Whilst those who are listed in room 2, please find the room next to this one to your left.
And this room will be separated. The room in the back will be function as the lounge for the participants to prepare for the presentations or to mingle with one another during the breaks. And the coffee and tea will be provided to your right of this room. And if you have any questions, our committee members will be available to assist. You can easily spot them with the bright yellow jackets, so you won't miss them. We would also like to inform that voting for favorite poster has started since yesterday and will be closed by tomorrow at 10. So cast your vote by scanning the barcode that you can find on the signage outside this room. And you can also see the poster directly as they are displayed on screen.
This is a great opportunity to explore the diverse and innovative research conducted by our fellow students. We also encourage all attendees to participate in the voting for the favorite poster award.
Thank you very much for your active participation and support in celebrating our young researchers' work. And we'll be seeing you tomorrow.
Wassalamualaikum warahmatullahi wabarakatuh.